Hi, my name is Jacob Kapner, and I'm going to talk about the role of affinity chromatography in the purification and production of vaccines. So vaccines are biological samples of viruses, such as inactive or attenuated forms of a virus, polysaccharides from a virus, subunits of proteins from a virus, etc. And they provide an acquired immunity to a disease. So the bigger picture and the societal impact of vaccines needs little to no introduction. They have caused epidemics to be controllable diseases in a matter of years. They reduce healthcare costs overall. They extend life expectancies for general populations, and plenty of other things have happened as a result of vaccines. Yet, in developing countries, 50% of the death rate comes from treatable diseases, many of which could have been prevented if a preemptive vaccine were used. So the question is, what causes this lack of a sustainable global supply of vaccines? And in part, the answer is this traditional purification train that you can see in the top of this figure. It is both cost and time intensive, has a lengthy series of steps, as you can see, with differential centrifuging, 2 anion exchange steps, gel filtration, solvent extraction, and it culminates into this process that is not meeting the demands of a sustainable global supply. So below, we have this more appealing model with affinity chromatography seemingly taking up 6 of the steps from the above purification train. I'm just going to go ahead and say this is a little bit reductive. Affinity chromatography can mean a lot of things, which the paper that I pulled this figure from knows and talks about depth, but we can get into that a little bit later. So the existing forms of affinity chromatography work on a lab scale for vaccine production and research. They are involved in the research for the West Nile virus vaccine, involved in the research for the coronavirus vaccine, and plenty of other examples. But large-scale production for viral vaccines with affinity chromatography has yet to be seen for a variety of reasons that I'll talk about in a bit. All right, so before I get into the existing affinity chromatography methods, I want to give a general definition of affinity chromatography. So affinity chromatography is a method that allows for the separation of a component from the mobile phase by a receptor-ligand-like interaction with the solid phase. So below, I have 2 examples that I think best show what I'm talking about here. So immunoaffinity chromatography has antibodies attached to the solid phase of the column that will bind to antigens going through the mobile phase. So you can attach a binding sequence for the antibody to a specific molecule so that you can pull it out of the mobile phase, or you can attack something that already has this binding sequence naturally. And then once you elute, you have a pure form of your product. So with a mobilized metal affinity chromatography, there is a nickel-2 ion chelated to the solid phase. And this nickel-2 ion can chelate to electron donor groups in the mobile phase. So for example, histidine is what is often used. Histidine can be attached to proteins, so a long chain of histidines called a his tag will be attached to proteins, and as they go down the mobile phase, they'll chelate with this nickel-2 ion and separate from the mobile phase. And then you can elute and get a pure form of your protein. So these are 2 methods of affinity chromatography that are useful to talk about because they have a set of pros and cons for vaccine production that is somewhat relatable to many other methods of affinity chromatography. But by no means are these the only methods of affinity chromatography. So the pros are somewhat obvious here. You can pull a compound of interest out of a mobile phase with relative ease and high efficiency. So also specifically with immobilized metal affinity chromatography, it has a relatively cheap ligand for the solid phase. And this nickel ion can also be swapped out for other transition metals based on the situation. Oh, excuse me. So that is a definite plus for immobilized metal affinity chromatography. Now I want to talk about some of the drawbacks and reasons why we don't see this for manufacturing vaccines. So I'll start with immunoaffinity chromatography. The cost is the major drawback here. The antibodies can cost several $100s per milligram. The antibodies lack the stability to be reused, so every time you use a column, you have to replace these antibodies. And finally, it has harsh elution conditions. So the solution you have to pass through to actually get your compound of interest has high acidity. So that can cause issues with your compound of interest, especially if it's a protein. So for immobilized metal affinity chromatography, it has several shared issues with IAC, but it also has some unique ones. So the proteases used to cleave the his tag in the post-dilution stage, that kind of further burdens the purification process, and on an industry scale, that adds another step, which is pretty cumbersome. Additionally, you have metal ion leakage. So anytime you are purifying something for a consumer product, you have to be extremely wary of potential contaminants, and this is a fairly serious one, considering nickel-2 is a carcinogen. So all in all, these are some of the drawbacks that have caused them to not be used on a large scale as of today. I could see these potentially being used in the future if the methodology changes a bit, or if there are adjustments to address these drawbacks. However, I am actually going to pose a different solution here to the question of what type of affinity chromatography would best suit vaccine production. And a novel solution that has been talked about is using molecularly imprinted polymers as a solid phase. So molecularly imprinted polymers, known as MIPs, create a receptor for an analyte by just using the analyte as a template. So you start with a template and add some functional monomers you know to be chemically active with this template. And once these monomers have bound, you have this complex that can be cross-linked, and in a polymerization reaction, it creates this solid phase here. And once you remove the template, it is a solid phase that can be used in chromatography. The paper that I actually got this figure from uses the MIPs in affinity chromatography to determine the concentration of antibiotics in milk, which I think is significant for a couple of reasons. One, it's an industry application and shows just how far we've come with MIPS technology in affinity chromatography. Two, it shows that it can be specific to the point of concentration determination, which is good because in milk, it could pull out many other different molecules, but it specifically pulls out the antibiotics. Okay, so this next experiment is a little bit more relevant to our ambitions of vaccine manufacturing, given that it results in purified proteins. So we start over here with this DYKD sequence, which happens to be the anti-flag binding sequence, or the epitope tag. It is surrounded by these functional monomers here that will bind to it, and EDMA is the crosslinker that will lead to the connection of all these monomers, and ABDV is the radical initiator of the polymerization. So if all goes according to plan, then these silica beads will be covered in molecularly imprinted polymers that are capable of behaving similar to flag and binding anti-flag sequences. So here you can see the SEM of these resins, and these resins are loaded into a column, and the crude cell lysate is used as the mobile phase. And here you can see some of the experimental design. They have an M-cherry fluorescent tag associated with the protein with the anti-flag on it. And this is so that they can record fluorescent signals in the elution to test how much of the protein is recovered using this methodology. So here we have a SDS page gel, and for those who aren't familiar, it is a method of separating proteins based on molecular weight. Here we have in lane 0, I'll call it, a ladder which has bands corresponding to different molecular weights. You'll see 96 kilodaltons here at the top, so on and so forth. And in lane 1, we have the cell lysate. So this is all the proteins within the cell contained all in this lane, and again separated based on molecular weight. More importantly, here we have lane 3, which contains our elution solution and our protein of interest, which is marked here by this arrow. So we successfully isolated our protein of interest from the cell lysate. This asterisk protein down here is actually something that comes out in regular affinity chromatography, immunoaffinity chromatography with anti-flag specifically. Apparently it is just an off-target effect of anti-flag, but it doesn't really matter because we isolated our protein of interest here to a high degree. So in conclusion, I believe that MIP chromatography has a high potential for large-scale manufacturing, but there really is no data to back this up. I'm not alone in thinking this. There are several review papers, including the review paper on slide 1 of this presentation, that think MIP chromatography is a promising solution to the issues we've seen with vaccine production. And this is in part due to MIP-based chromatography's ability to purify and recover molecules at such a high rate. So what I didn't mention in this last experiment here was that the fluorescent quantification of the protein in the elution versus the residual mobile phase is that the recovery rate was as high as immunoaffinity chromatography. So in essence here, they've created a chromatography that effectively mimics immunoaffinity chromatography in effectiveness while maintaining a lower cost in the process. On top of that, you can reuse these columns and you don't have to replace the resin every time you use it, which is incredibly convenient because I'm sure that polymerization reaction would be a huge drawback to have to redo every time you did one of these on an industrial scale. So we've essentially kept all the pros for immunoaffinity chromatography and removed all the drawbacks, which is incredibly promising and could lead us to a similar purification train as what was proposed in review paper 1. Again, this doesn't capture all of it, but it is something we can aim for. So I chose MIP-based affinity chromatography not because it is the only new generation ligand that could potentially be a solution here. There were several other new generation ligands that could also potentially be a solution for vaccine production. But I think MIP-based affinity chromatography is by far the most interesting and may have the most applications in other fields. That's part of the reason why I included the milk antibiotic paper, was because it's just an example of something that is kind of not what you would initially think of, but an application of MIP nonetheless. I think that the idea of printing a receptor based on your analyte is incredibly powerful. And so that's in part why I chose to talk about this. Also, I would just like to talk about the fact that in 2009, there was a paper written on MIP technology at the time that stated MIP was no longer capable of, or not no longer, but MIP was not capable of isolating products above the molecular weight 10,000, or 1,000 rather, excuse me, grams per mole. So I just wanted to point that out because we have come a significant way in the last 10 years, because we have been able to isolate a 32 kilodalton molecular weight protein. So that is to say that we could, eventually produce a large-scale manufacturing process with MIP-based technology for vaccine production. So that is the hope. Thank you for listening. And if you have any questions or comments, Please leave that in the Canvas comments section. Thank you.